So can we begin, Tom?
Yeah, go ahead. It's 2 p.m.
Okay, hello everyone. I'm Anusha from CSA Attest India, and I'll be moderating today's talk along with Tom, who's from Utrecht University, Netherlands. So today, as a part of SNEV, EV characterization series, we have a Professor Dr. Michel Pfaffl, who will be talking about RNA sequencing and EV biomarker in molecular diagnostics, mainly focusing on the MIQE guidelines. So before we move to the session, I'll just briefly introduce Dr. Pfaffl. So he's a professor of molecular physiology at Technical University of Munich. He's principal investigator at Institute of Animal Physiology and Immunology. His work focuses on gene quantification, RT-qPCR, RNA sequencing, extracellular vesicle biology, etc. And he's also the co-author of MIQE Guidelines 2009 and MISIV Guidelines 2018. So with this brief introduction, we can begin the talk. So just before starting, I'd just remind the audience that we can, you people can post your queries in the chat box. We'll have a Q&A session at the end of the talk. So with that, over to you, Dr. Pfaffl.
Thank you very much for inviting me here. So I was invited in Melbourne at the last ICEF conference, and it's a pleasure for me to present my work. I hope everybody can hear me very well. Otherwise, I speak a little bit louder. And yes, it should work. And I was asked to introduce you into about qPCR problems. And I thought this is a little bit too less. So I expanded my talk in the first 10 minutes about the MIQE guidelines, and the rest of the talk, another whatever 40-50 minutes in what we are really doing. We are using, we used to have a RT-qPCR for gene expression profiling in various of samples, and now we are moving over for next generation sequencing with focus on small RNA sequencing, especially microRNA quantification, biomarker discovery in EVs, and that's why I'm here invited. But the first, so going like this. So disclosure, I'm just a professor at the Technical University of Munich. I'm not connected to any companies or something like that. So all what I tell you is free and is free knowledge, and almost you find all the up to millions or even billions of molecules in your biological samples. Okay, and this also should be covered within your quantification procedure and is also one of the hallmarks of the assay validation. LOD and LOQ stands for limit of detection and limit of quantification. So limit of detection is more or less the sensitivity of your assay. What is the lowest concentration you can quantify? It's the limit of detection, but it's not the same like limit of quantification. The limit of quantification is a little bit higher level. In the PCR assay, more or less about between 20, maybe and 50 molecules. This means the limit of quantification is defined also as the lowest limit of what you can quantify, but with a high reproducibility. So normally you test the limit of quantification, you set up 20 replicates with the same amount of molecules, and the lowest quantification you can measure in 19 out of 20 samples, means with a confidence value of 95%, then you are at the point of limit of quantification. So this all sums up under the PCR problems, or more or less what impacts the variability of PCR. So even after the reaction, we also have points which really impact our results. And this is how I handle my data. You know, a PCR, they will always have a kind of exponential curve in quantitative PCR, but all these raw data, what you actually get from the machine, are not reflected on the graph you see later on. So all these kind of amplification histories, these exponential amplification models you see are all smooth. That they don't reflect real scientific data, meaning every machine, every PCR cycler are smoothing, are rounding these data, are making mean values and something like that. So if you really want to compare different assays on different platforms over different labs or even countries, you have to go back to the raw data. And remember, going back to Corona crisis, this was exactly the problem we had, that some people reported crossing points, other people reported molecules, other people reported different standards, but nothing was really the same, okay? So therefore, if you really want to compare different methods, you have to go down to the raw data, which are not impacted by rounding, smoothing, curve algorithms or something like that, which are all implemented in the real-time PCR cycle, okay? This, and this also goes back to the background correction, what you subtract as a negative sample and something like that. This was a huge matter of discuss during the Corona crisis, because different countries, or even within Germany, we had problems with different labs and something like that, okay? So then also how to handle outlier, this is also a big question. Also, a lot of problems occurred in the Corona crisis. Okay, but then also the red or the pressing topics is the normalization strategy. You all know, if we are doing a gene expression over gene over mRNA, it's always important to normalize it with a grown housekeeping gene, or now we call it reference gene, which should be not regulated under the treatment you performed. And this was developed by Giovanni Sample early in the 2000s, 2002, I think, was a genome publication. And he said that one reference gene is not good, use multiple reference gene. He suggested to use 3 non-regulated reference gene as normalizer. And I think these 3 reference gene are now the standard for a good normalization, or let's say comparison of different biological samples, okay? And to choose which are the best reference gene, there are some tests developed, like Genome, BestKeeper, or NormFinder are the most usable tools for that, to test which are the best reference genes in my assay, in my evaluation, in my study cohort. Okay, so let's come to point 11, which what introduces as well a lot of variability, is of course qPCR efficiency. What is qPCR efficiency? Normally we learned at school, or you learn at university, that in every cycle the DNA will be doubled. No, this is not really true. This is actually bullshit. There is never a perfect doubling of your DNA. In theory, there is a perfect doubling, but in reality, it's something between 90, 95, or often below 80%. And if you calculate 2 by the power of whatever cycles you perform, let's say 30 cycles, if the curve goes up, and if you calculate only 1.8 by the power of 30 cycles, then you end up with totally different values. So we introduced then in 2001 the qPCR efficiency correction into these mathematical models, and this changed more or less the quantification of PCR dramatically. Last points are then, of course, the statistical test at the very end, which is very important for the interpretation of your result. But if you're having a good statistics, a good experimental design of enough replicates, then it's also fine that you can apply a good statistical test later on. And if you have a statistical answer, is it regulated, is it non-regulated, you have also a biological relevance, and you have a significant more or less gene expression change after your induction. So that means there are quite a lot of problems which interfere in the PCR reaction, pre-PCR or post-PCR, and that's why we were sitting together 2008 and 2009, these 12 guys. It's long ago, it's already 15 years ago, and you see all these guys, you even see me here with hair on my head and something like that. And all these guys were sitting together, international team, a little bit focused on Europe, from Czech Republic, Belgium, England, Germany, US, Sweden, and so on and so on. And all these guys were developing the so-called MIQE guidelines. And these are the 12 MIQE authors. And what are the MIQE guidelines? Of course, the MIQE guidelines is more or less the big brother of the MICE guidelines, or let's say the initiator of this MICE initiative. It's a set of guidelines that describe the minimum information necessary for evaluating the qPCR experiments. And why we set it up? Exactly from that reasons, because in literature there was a lot of debate on how to perform PCR, how to interpret it. But the real goal was to provide guidelines for the authors, for the reviewers, and also for editors, which are reviewing your papers and are editors of the journals, to get a clue what is important in these reactions. And qPCR is now a standard method. It's running in every lab, every molecular biological lab, and therefore it's very important sorry, that all of us are on the same line. That's why authors, reviewers, and editors. We want to establish a clear framework to conduct the experiment. Of course, this will introduce experimental transparency and also the reproducibility. If you know exactly what you've done, since you have to know exactly what you're doing, then you increase your reproducibility in your lab, and also you can transfer these. What, can you still see me? Because I got then your screen wurde unterbrochen.
Oh,
Hello?
yeah. We're still
Yes,
we're
we can
stuck
see.
at your first slide, I
You
think.
still can see me, okay?
Yeah.
So you can
Yes,
still
yes,
see
but
my
the slides,
Slides.
yeah, are not moving, I think.
Okay. So because here I got a message from the IT. Okay. So 15 years of MIQE. So Stephen Bastin summed it up in a new summary in molecular aspects of medicine. And he said that the MIQE guidelines have played a significant role in promoting transparency and reliability in all kind of labs, and that they are shaping the landscape of gene expression analysis and in any kind of all kind of PCR related quantitative methods. But this field is still evolving, and therefore we are working on an update, and the update is under production, and it will be published in autumn 24, though there will be a kind of MIQE 2.0 this year, later this year. Okay, this was kind of the beginning, a little bit problematic in technical. Okay, since the technical was not good enough, but I hope now the second half of my talk will be much better. And this is more or less how we get from RT-qPCR to small RNA sequencing for microRNA biomarker discovery in EVs. And this is actually what we're doing today in my lab, looking for biomarkers in circulating nucleic acids. And the first question is always, do we have a free cell-free transcriptome in our circulation? And since the blood is the major topic or major source of our liquid biopsy and nucleic acid, yes, there was a very nice review which is depicted here, and they showed that we can find any type of RNA, of course, mainly blood-based types of RNA, but also any kind of organ or tissue RNA we can find in the blood circulation, as we call it cell-free RNA. And we can use these cell-free RNA within our liquid biopsy. So liquid biopsy, you all know, it's blood, urine, saliva, stool, milk, or whatever. And we can use in these tissues different types of targets. It used to be circulating tumor cells. It used to be a lot of circulating DNA. And what we are doing, we are focusing on not only on microRNA, we are focusing more or less on any kind of RNA species in EVs, in the liquid biopsy diagnostics, which we can use later on for diagnostic, for prognosis, for personalized medicine, and for disease monitoring. I think I can a little bit skip this EV and exosome stuff, since we are all aware of EV and the importance and how we can use it. But you know, a lot of publications are coming out, also in the biomarker scene. And also I can skip this slide, which tells up which vesicles are important. I adapted this from Marka Wauben, from one review she wrote. But I think we are all EV people, and since I can skip these EV more or less information. Also says, which tells us that we have a lot of different analytes connected to the EVs, and in special the nucleic acids. We have microRNA, mRNA, and DNA. And some publications tell us that they are protected and are in the inside of the EVs, but today we know that we also have a lot of binding of nucleic acid to the outside, to the corona. And this brings us to the next slide. We know from a lot of publication that show us that the EVs provide a protected source, especially for microRNA, meaning the microRNA is protected by the membrane, by the EV membrane, and therefore we get much better results if we first prepare the EVs and then isolate the nucleic acids, and this gives us much better diagnostic results. What is the meaning behind it? Why the organisms are doing it? So they exchange the so-called extracellular RNA, the xRNA, between the cells. In the focus is definitely mRNA, microRNA, and other RNA species like long-coding RNA, and they use it for information exchange. But how does it really work? It means at the beginning we thought that these RNAs are, or that these nucleic acid in the whole family are intrinsically stable in blood, in these urine swabs, in these liquid biopsies. But this is definitely wrong. Any kind of free RNA is fastly degraded in the circulation, and that's why we need a type of protection. And this protection is either means that these extracellular RNA are bound to proteins or are protected inside the vesicle. I think this is proven, and this is proven by a lot of publications. And these vesicles could either be apoptotic bodies, or here you see the argonaute as our binding RNA binding protein, or classical exosome or V protection. But we also published some paper where you have the RNA stored in HDL associated vesicles. And there is also a new idea born a couple of years ago, 2 to 3 years ago, that the circulating RNA is also associated to the so-called EV protein corona. What is the EV protein corona? It's actually just the region around the EV. So the EV is not naked, which is kind of the old view. A pure, a naked RNA is a good EV. This is not true. If you want to have a functional EV, it needs an intact corona, and the intact corona binds other proteins, binds a lot of nucleic acid, and especially the longer the mRNA, the DNA is today seen, which is bind to the outside. We are involved in studies doing the quantification of these models. These graphs are taken from the University of Salzburg. Dirk Strong's team is working on that, and we're working with them together to do different EV isolations and looking together with corona, without corona, what is the RNA content without corona. But I think this is a new idea, and it's still a matter of debate how it works, how the binding is promoted. We know it's working, but what we don't know so far is how a good corona is generated, and how we can keep the functional corona during the extraction. We have some ideas so far, and especially for the functional people, for the functional EV people, it's very important to have a functional corona. which are uptaken by the recipient cells, the EVs, and of course the microRNAs. And this brings us to more or less a picture of different different uptake, a mixing of the EV in the cell. So EVs are released from the cells. They travel through our circulation and are uptaken, and you see here the blue and the red vesicles, and they are mixed in the cells, and then they are released again. So we have a kind of high rate of exchange of EVs in our body, and so far we don't know where all the EVs are coming from, and we have no idea where they are really going to, and which cell is uptaking which EVs in which content, and so on. So, and the meaning, coming back to microRNA, is of course that they exchange the RNA from one to the other cells, and within the cells, then of course the RNA is implemented in the RISC complex, and the RISC complex is the RNA-induced silencing complex. You know it from your school or lectures, that the RNA is uptaken and binds to specific, with the seed sequence, it binds to specific target sequencing of mRNA, and if you have a perfect match, you have a degradation of these RNA. Therefore, we see that the microRNA is a regulative tool in the target cells, which can break down specific mRNAs in the target cells. So then going backwards, why we are talking about vesicles? Okay, we heard about that this is a protected area for all the nucleic acids, but this has to be proven. So in the beginning, we tested different biomarkers in cells, in serum, and then also in EVs and exosomes, which were isolated from the same compartment. So we looked up of blood compartment specific biomarker signatures. We have done these in sepsis patients, and our first question were, should we take full blood? Should we take blood cells? Should we take cell-free compartment, or should we take any kind of extracellular vesicles? And if we are thinking about extracellular vesicles, we can also think about, should we take exosomes, microvesicles, ectosomes, ectomeric, apoptotic bodies, or just all extracellular vesicles? So all these questions are have to be answered, and that's why we set up an experiment from sepsis patients, where we got 14 patients, where we isolated from the same amount, always from 1 milliliter, we isolated exosomes, now we call them EV, a serum, or just everything what is in this 1 milliliter. We extracted the RNA from these 3 compartments, and then we did next generation sequencing, and from this small RNA seq, and from this I will show you the results, and you clearly see if you have a PAX gene, you have much more cells, you have much more mRNA, you have much more information, and that's why the mapping is much better. But if you're looking here for the total serum, and for the exosomes isolated out of this serum, we had much lower read counts. But if we expand it, if we make a relative mapping, you see that the exosomes or EVs have an advantage over classical serum. You have here the protection, you have here the enrichment of the factors 3 to 5 is always our internal counter of the information. And therefore, if you isolate EVs out of blood compartments, you get a concentrated information, which is comparable to kind of the same information you get from PAX gene from this blood. Then we analyzed these samples. This was just a trial study to see where are the advantages, why we should use exosomes. And here is depicted from one and the same blood sampling. If we analyze these compartment-wise, we see that we have, these are sepsis patients compared with healthy controls. We see that we have different biomarkers in the PAX gene, in the cells, more or less, compared to the exosomes and the serum. Of course, the exosomes are part of the serum, therefore they share the majority of this biomarker. But these are 2 different biomarker signatures. One is cell-specific and the other is kind of circulating specific or cell-free specific. And if we pick even these biomarkers, which we hear in this first trial experiment, healthy volunteers versus sepsis patients versus septic shock, meaning severe septic patients, even single biomarkers can predict even here in the seconding if the people survive from a really single biomarker perspective. And this showed up that EVs in sepsis, in pneumonia, in critical illness might be a good biomarker for testing at the first day of hospitalization. And that's why we decided to continue with EVs and microRNA as an early biomarker in pneumonia, sepsis, and severe lung critical diseases. Okay, and over the years, we developed more or less a kind of framework, which are the advantages of the microRNAs, and I summed it up here. Yeah, the advantages of EV-associated microRNAs are that they are taken from liquid biopsies, which are seen as non-invasive. MicroRNA are short molecules, 22 MERs in these regions. That's why they are stable. Furthermore, they are protected. We've seen this. They are protected by the EV. They are protected by the corona. So we have a very stable biomarker. And since these EVs, microRNA, are then already amplified. The signal is amplified. I showed it 3 to 5 times. They are amplified in the EV. Then we use this expression change for an early evaluation of physiological changes. And with the combination of various microRNAs, we come up with a biomarker signature. And the advantages of a biomarker signatures is that a biomarker signature consists out of 10, 12, 15. This is a range where we are working in different diseases, are specific information which can trace back to single diseases, to single events in the patient, and therefore they are a physiological or pathological indicator. They can be directly related to a specific disease, a cancer stage or a cancer subtype. We are working as well in breast cancer, in head and neck cancer, and other cancer types. And the second big advantage is if you have a microRNA, mRNA, or something like that, you can afterwards amplify the signal, meaning the signature is amplified later on by RT-qPCR, and therefore you have an easy and early detection because you have a first amplification of the signal in the EV and the second amplification by RT-qPCR. And on the other hand, we can standardize everything via MIQE or MyCF guidelines, and therefore we can have a specific and reproducible result at the very end. Good. This is our workflow, how we've done it in patients. So we take the patient, we sample the blood, we purify the EV, we characterize the EV. I will come to that in a couple of minutes. Then we prepare make libraries, we prepare the RNA, making libraries, we do the RNA sequencing, and then with a lot of bioinformatics, I will focus on that as well, and validation via qPCR. We have given RT-qPCR and a microRNA signature for a specific disease. Okay, so we sum this up under microRNA-based decision-making. We have to do this, or you should do this in 2 cohorts, in 2 steps. In the first cohort, where you learn, where you develop the signature, where you classify the patients. Here you see some examples from sepsis patient in a couple of minutes. And of course, you have to physiological and functional validate your result. And in the second cohort, you confirm your biomarker signature. And this in a little bit more detail, how we've done this, or how the workflow should be. So as this was published, and we've done this in a training set of 67 patients. First, of course, we sample the blood, we do the EV isolation, we do the RNA extraction. After that, we have the library preparation, the small RNA sequencing. And then, of course, complex bioinformatics, multivariate data analysis, where we analyze differential gene expression using PCA, PLSDA, or SPLSDA. Yeah, I explain it later on. In between these steps, we have always quality control steps, meaning after the EV isolation, we have the EV quality control, the MIQE guidelines. After the RNA extraction, we have RNA-qPCR, more or less MIQE guidelines. After the library, we have library prep QC, which is more or less done retrospectively from the next generation sequencing result. Then, of course, we have the NGS data QC, and doing then bioinformatics, establishing first biomarkers. Then we validate these biomarkers first in a RT-qPCR experiment. And then we have the candidates. Since we just have a first round of our patients, and then we need independent patients, we need independent biological samples to validate these samples then in a second cohort. Okay, I will show this all in a minute to you. So yeah, I also skip this because we already have 45 minutes. You know about the MIQE guideline. Everybody heard about a lot from Ken so far, so I will skip these. Yes, and then we coming to more or less our results. Since we know now that microRNA are good in sepsis diagnosis, we want to establish a fast and reproducible methods which is reliable in the daily clinical practice and which is suitable for microRNA-based biomarker discovery. And then we had the problem, yeah, which kit should we use? Normally we're using ultracentrifugation. You know, this is the state-of-the-art method, but of course in the clinics it's useless because it runs overnight and no clinician will do it. So we need easy applicable methods in the lab like Exicon, QEV, XORN-easy, precipitation, or membrane affinity methods. And that's why we benchmarked all these methods in 10 sepsis patients and 10 healthy volunteers and compared it with the so-called gold standard ultracentrifugation. We've done this, okay, here more details. We published this in the JEF. If you want to see all the details, just a short thing. First, we characterized all the EVs, MIQE compliance, of course. We've done these different isolation methods, and of course, then we isolated the EVs, we concentrated or resuspended them, and then we did surface marker by Western blot, we did NTA, size distribution, and of course, TEM. Today we are doing cryo-TEM, but during these days we started with transmission electron microscopy, and then we applied this to these to different kits, to different isolation methods, and compared more or less the diseased and the healthy patient by a next generation sequencing, and compared more or less the result between the diseased and the non-diseased. All was done from the same patient, always 1 milliliter, okay? And what we compared healthy volunteers with people coming to the ICU at day 0. So this was a day of hospitalization. We are doing our analysis, and then the clinician want to know, is there a high severity of sepsis? How is the survival rate of the patient? And will these results help me in the diagnosis of this sepsis patient at hospitalization? Some slides about these characterization. Here are some TEM pictures. And on the left-hand side, you see, of course, the results of these different isolation methods. And you clearly see different isolations gives you different subpopulations of the EVs. QEVs and ultracentrifugation and so on here in these box plots will isolate different subpopulations. And you can think different subpopulations of EV, even if they're different in size, have different biomarkers bound to the surface or protected in the EV. And this is also depicted here in the readout of the mapping efficiency. Some have a higher percentage of mapping efficiency in reads and others are lower, meaning that some isolation methods are more usable for next generation sequencing and others not. And we summarized it here in a kind of a kit ranking, also going a little bit deeper into the topic for RNA impurities, and meaning like short reads comparing to microRNA, like ribosomal subunits compared to microRNA, or tRNAs compared to microRNA, which are more or less impurities. And you see different isolation. We have a classical lighting system. Green means good, red means not so good. And you clearly see that some methods like mercury or ultracentrifugation are more or less the best methods for microRNA analysis. Very important. We are just talking about microRNA diagnostics. This has nothing to do with proteomics or something like that in terms of microRNA readouts and purity of the retrieved RNA. Therefore, microRNA is the best in our perspective. And then grouping this, making here different clustering. You clearly see that you have two main clusters. One is the mercury, the ExoRNA kit, which cluster very strongly to the what I said, microRNA different certification cluster in one cluster, and even the samples, the patients cluster in between, and the other cluster is consisting out of exospin and QEV, showing us that there is a significant impact on the isolation on the later microRNA signature. So, and this is the impact of EV purity or EV corona. Of course, we have to work these out, but take-home message is every isolation method has its advantages and disadvantages, and we are going more or less for the microRNA isolation or ExoRNA for our RNA analysis. And if we are talking about microRNA biomarker signature development, we have to look what are the possible biomarkers, and therefore we are first looking on the significantly regulated microRNAs between the healthy and the possible septic patients at day 0 at the hospital. And you clearly see the more readouts you get from your biological sample in mercury, ExoRNA compared to exospin or QEV, the more candidates, significantly regulated candidates you have. Here you see here, if you sum up mercury, you come up in whatever to 250. If you sum up here QEV, you come up maybe with 40, 50 microRNAs, and making out of these whatever significantly regulated later on a biomarker signature. It's much easier to perform these or to generate these from mercury or ExoRNA candidates. Okay. Nevertheless, validate your resamples, validate your first candidates. Here we validated the 10 most up and the 10 most downregulated microRNAs. First, the NGS results are the red one, and then we validated with this RT-qPCR, and you could see that the correlation is about 95%. Always very important what you find in NGS. It's often not present in PCR. Here we were quite lucky, but you see that the dynamics of regulation is quite different, but at least the strength and the correlation over these two quantification methods is given. So what we developed so far, we developed a standard pipeline for small RNA. What I've not implemented in the talk are tRNA halves or piRNA. What's not mentioned here is long RNA. What I will show you in a minute are multivariate analysis. You just seen a heat map, but you will see PCR results very soon, and also the in silico validation. But for that, we need a pipeline. And this will be shown now. We called it Carnage, and the paper is more or less on the way. That is Carnage pipeline, and we also developed other tools like MyRef, with every PCR reaction, you need to have stable references, and we use our knowledge from the old qPCR times, and developed based on these algorithms also stable references in EVs, called these MIREF, for microRNA references in EV. And also what we implemented in this pipeline is isomeral. We are not analyzing the microRNAs. We are also doing isomer detection. And what we are working on is the transcriptomic network. So let me just introduce you to the Carnage, how we've done this, why Carnage, so the French pronunciation, which sounds a little bit more elegant. Comprehensive analysis of small RNA gene expression. So far, we're focusing on small, but the pipeline will be elongated as well to mRNA and long non-coding RNA in the future. For now, it's small. Obtaining valid biomarker signature from the EV made easy. What we are doing, we are just not focusing alone on the microRNA. We are all focusing on all RNA subspecies. So we import the raw sequencing data as a study design, we trim, we align the tRNA, and then probably we got one readout as a tRNA half fragments. This is one very important information, because also tRNA halves have a diagnostic value. I will not mention it further on here, but you should know it. Then we align to the microRNA, and further on top of that, we also evaluate all microRNA isoforms. Also not matter of debate here, this is another talk. What is the advantage of microRNA and their isoforms and diagnostics, okay? And then we do a total transcriptome alignment. So in the end, we come up with a focus on microRNA, on tRNA halves, and all RNA classes, okay? That means this is a classical readout. The normal or classical analysis just focuses on microRNA and the rest is rubbish. No, we use all the other subfamilies, and we could potentially use them for biomarker purposes. At the second focus, as I said, is the microRNA, the isomer families, and the tRNA half, which you are working on more intensively on. So the output of this developed software pipeline is multiples. You fill, at the end, you will see 5 boxes. First, always quality control. So this tool can also correct for batch effects, since you get samples from different hospitals, from different isolations, and something like that. This can be taken into account, and these batch effects can be subtracted from the sample to make them more comparable. Also, the detection of outliers. If you have some specific outliers, this software will detect it, okay? I just highlight now on the blue stuff, which is most important. Then, of course, we are going for differential gene expression of the candidates with the statistics behind it. This is more or less a DGE. And of course, we focus as well on the non-regulated microRNAs, because these are very good candidates for normalization. And in every let's say, in every good RT-qPCR experiment, you need a non-regulated reference gene, and she should be from the same family you're analyzing. Therefore, we cannot analyze with whatever small-RNA or something like that. We have to use a non-regulated microRNA in a microRNA biomarker study, okay? Very important. Then, of course, focus is on isomer analysis. We also analyze all isomers. And with these analyses, we can, in some cases, we can double the amount of biomarkers. We can double the amount of biomarkers with all the isomers, which are all, in other studies, all forgotten, okay? And we see, due to these isomer analysis, are the microRNA are expressed in clusters, how they are localized, and in which motifs they appeared. Okay, and at the end, we can cluster, we can classify the biomarkers via various bioinformatical methods. We can classify the biomarkers. These can be different sepsis stages, what you see in a minute, or different cancer stages or something like that. And at the very end, we also do enrichment analysis, overrepresentation analysis with gene ontology, CAC, and so on, to see will this biomarker pattern is there a physiological relevance? In which pathways are showing up? And are they kind of, are these pathways then connected to the disease I'm looking? Okay, good. The pipeline is still on our server. We've analyzed so far, don't know, dozens of studies or something like a cancer, a sepsis, even urine, blood analysis, whatever. We have a lot of different studies analyzed with it, and it's always optimized.
Sorry, in the same time, I think we need to start wrapping up to allow some time for questions.
Okay, I have another five slides.
Maybe a bit much.
Yeah. Should I end or will the talk automatically end after 1 hour?
No, not that, but we scheduled until 3 and then have some time for questions. So
okay, let me another five slides and then it should be over. Okay. Okay. So maybe I skip this biomarker selection and at the end to show some results. We could find a biomarker signature based on 12 microRNAs for different types of community-acquired sepsis, pneumonia, sepsis, and healthy volunteer. And based on these 5 biomarkers, we could reach in the training set accuracy of about 92 to 95%, and in the validation cohort of 73%. And for the first shot, it was quite good, we thought, and therefore we are continuing and also sampling more cohorts. And we presented this to the clinicians, and they said, yes, fine, nice, 12 microRNAs, but can we drop these down to maybe 3? I said, wonderful, yes. So, of course, the advantage of a signature is to have more more biomarkers in one signature, which is makes them much more stable. And then we condensed it to 3 microRNAs, 193, 542, and 1246. And even this gives you a good confidence. As you see here, we just making a mean value out of these microRNAs to distinguish between healthy patients, community-acquired pneumonia or a slight sepsis, more or less, and a severe sepsis. This is the most important, what the clinicians are interested in. All is published. After that, we validated our biomarkers in the physiological way. This was all done by Kanash. So we are focusing on cellular and humoral immunity, antimicrobial response network, and pathogen influence signaling, and this all fits very nice to sepsis and pneumonia. And all this is kind of summarized in a review which we published early this year, a pipeline for the development and analysis of extracellular vesicle-based transcriptomics in molecular diagnostics. And this is more or less all I showed to you explained in detail, okay? More or less the talk summed up in one big review. Where is the future? In the future, we also want to implement long non-coding RNAs as a transcriptomic network. We are working on that, but since long non-coding RNA are not that defined, that intensive like microRNA or mRNA, it's a little bit difficult, but yes, with the future, I think we will succeed. So this is more or less the summarizing slide. At the beginning, we have training cohorts where we sample the blood, where we do multi-omics, where we do biomarker selection. Today, I just showed you the microRNA analysis. We're doing much more. I mentioned mRNA long non-coding. We are also doing tRNA half. We're doing also proteomics, which we want to involve into multi-omics study. Afterwards, you need to validate these biomarkers in a second study. And finally, I hope that then everything comes in a third cohort, or it comes to the routine clinical testing. So acknowledgement for the biomarker studies, it's Dominic, it's Benedict, and so on. We collaborated with our university hospital, with the City University Hospital, Klinikum München-Perlach, and also the LMU clinics. And since it was supported by the Ministry of of Economics and Energy, there are always companies inside. I also mentioned the Universitätsstiftung and the Bayerische Forschungsstiftung. And also important for you, you can see here these QR codes where you see all our publications which are connected to the MIQE guidelines and advantages of the MIQE guidelines, or to EV biomarkers. So we already have 35 papers on that. And also here, the access to all these software tools, which I show to you, all is open and all is free for download and use. And at the end, I want to end and thank you for your attention. And you see, we are also kind of very actively on national and international meetings. And with that, I want to thank you, and I'm looking forward for questions. Thank you very much.
Thank you so much, Professor Pfaffl, for sharing your knowledge with us. We go to the questions. First question is from Nada. I'm not sure if you want to unmute yourself and ask directly. Otherwise,
Hi, yes, of course, no problem. Yeah, thank you very much. I just wanted to ask, just because, as you mentioned, when we get our NGS data for microRNAs from EVs, the problem is a lot of them would be, significantly, even the significantly differentially expressed would have a really low TPM as like transcripts per million. And I was just having this question of what would be detectable by PCR? Please.
Very good question. So we have the threshold with any kind of biomarker we are working with is 50 reads. 50 reads is just to have it in all your analytical tools. And to be honest, in RT-qPCR, you need at least 500 reads. In the range of multiple hundred reads, 500 reads and more, then you also find the candidate later on as well in PCR. If it's a good microRNA, often you have isoforms, and even this is also not working. But I would say 500 plus is a relative good candidate, yeah, which is also working later on in an RT-qPCR reaction.
Thank you. I mean, in EVs, I'm unfortunately, from what we've been seeing from our NGS data, 30 is like our more or less the maximum TPM we're getting. So you would recommend that probably might not show up in PCR, right? Thank you for that.
That's right. But we do the cutoff very strict at 50. Very strict at 50. Of course, we have studies. We have one study in asthma where we have lavages, with a very very critical, I have to say, very critical. There we go down to 20, 30. But we can publish the paper, but we really have problems to get real biomarkers in PCR after that. Really. So I would recommend 500 for a good biomarker. So we are not going in a biomarker development lower than 500.
Thank you very much. And in terms of the kit for PCR, like as in, the one that would have the most sensitivity for EVs specifically, would you recommend one, please?
It's not the kit. It's not the kit. It's your work practice. If you have a good work practice, if you have a good isolation method, if you want a good EV isolation and later on a good RNA extraction, any kit will work. Since in the study I presented, since we are working with Exicon, during that time Exicon was an own company, then it was taken over by QIAGEN. We didn't know this. But this turned up that this is kind of the best kit. But let me tell you, this is also a matter of the corona. Since the mercury kit is a precipitation, a simple precipitation, and the QIAGEN's kit are affinity-based kits to the membrane, they have a protected corona, meaning these are, we call them crude EVs. They are not the pure one. Years ago, we were go for pure EVs. There are no corona, and not a precipitation. A precipitation is bad. Yes, it's not a pure EV. But if you isolate and produce pure EVs, you get rid of the corona. And as soon as you get rid of the corona, you're losing a lot of biomarkers, because a lot of biomarkers are sticking outside the EVs. This is the corona ideas. And since 2 years, when Bernd Giebel and also Dirk Strunk showed, pure EVs is nice for characterization, but they're not working at all. There is no functionality. They need this corona around to be functional and be uptaken. Then more or less the EV is in the focus, and also then the methods leaving an intact EV. And these are more or less the not richest method, which isolation method which come up with, of course, high microRNA concentrations and also high amounts of contaminants. So let's do it like this, okay?
That was very helpful. Thank you. And my last question is about the normalization genes for microRNA in human plasma. Again, would you kindly recommend any from your experience, please?
Again, which microRNA I recommend, or which method to
Which
evaluate?
microRNA, maybe, please? Which microRNA for reference genes?
Good question. Just go to the MyRef paper. Just go to the MyRef paper. I can't tell it by heart now. There we show 3 microRNAs which we use for normalization in the PCR assay. I can't tell it by heart now. And therefore, we had 12 studies, different, and we selected them by different normalization in the next generation sequencing, different normalization in PCR, best keeper genome and norm finder, and brought these together. You can read it up in the paper, which are good for human blood-derived EVs. Okay, this is the only this is the only one you can really validate, because there is the only really base where I have enough publications and enough data available to do all this evaluation. All other cannot be done so far, because you don't have enough the papers in urine and so on where you get all the data from.
That's true. Thank you very much. I really appreciate it.
Thank you.
And thanks a lot. There were more questions, but I think in the sake of time, we need to wrap it up. Can we still send these to you via email? Because
no problem.
Looking for answers. Great. So that concludes this session. Thank you, Professor Pfaffl, again for preparing. Our apologies for the small hiccups that we experienced, but it was a really nice talk. So thank you very much.
You're welcome. Thank you.
Thank you, Professor.
Bye-bye.
Have a good day, all.